Now look at this.
What's a bit about penguins? Today we do topic 5.2, meiosis and genetic diversity. I accidentally did a bunch of this stuff during 5.1 when we talked about meiosis. There's a lot of parts of meiosis 1 that are going to lead to genetic diversity. So we're going to kind of focus in on those steps of meiosis that really lead to all this genetic diversity. So during prophase 1 of meiosis 1, this is when we saw the chromatin condensing and the homologous chromosomes were going to find each other and make themselves into that beautiful little tetrad. This is when we're going to see crossing over taking place. So as we said before, the non-sister chromatids are going to exchange genetic information, and that's going to lead to recombinant chromosomes. Recombinant chromosomes are chromosomes that are going to have two different organisms or two different individuals' genetic information together. So this is considered a recombinant chromosome because you see we have mostly pink and then a little bit of purple. And here we see mostly purple with a little bit of pink. So it's a combination of two different parents' genetic information on one chromosome. So during metaphase 1, we see independent assortment. Independent assortment has to do with homologous chromosomes are independently aligning on the metaphase plate. In this example, we're just looking at an organism that has two chromosomes. And so as you see over here, we've independently assorted so that all maternal was on one side and all paternal was on the other. And then in this case, we see that we have paternal on the left, maternal on the right for chromosome 1, and then it's opposite where it's maternal on the left and paternal on the right for chromosome 2. So based on how these homologous chromosomes are lined up during metaphase is going to lead to a difference during anaphase. So during anaphase, the homologous chromosomes are going to segregate and move into opposite poles. Once they segregate and move to opposite poles, that's going to cause the daughter cells to have this new combination of genes. They'll have some maternal and some paternal chromosomes within that one cell. So based on independent assortment, looking at this example, there's 4 different options. 2 to the 2 is 4. But in our case, we have 23 different pairs of chromosomes. So 2 to the 23, 8.4 million different combinations, which 8.4 million combinations is definitely genetic diversity. And the last way that we see genetic diversity has to do with random fertilization. Any sperm can fuse with any egg. So since the sperm have 8.4 million combinations and the eggs have 8.4 million combinations, based on independent assortment alone, that's 70 trillion different combinations. You are pretty unique. So I hope that was helpful. Remember to hit that follow button because I'm doing this for every single topic in the CED. AP Pain was just success. Bye, y'all.